be looking at Chapter 9, which is titled From DNA to Protein. The beginning of our chapter talks about an interesting concept about ricin. It's amazing how something naturally occurring like that is so highly toxic. It says a dose as small as a few grains of salt can kill an adult. Only botulinum and tetanus toxins are more deadly, and there is no antidote. Well, it turns out that ricin is actually a protein, and we've been studying DNA lately, and DNA, as we know, is the instructions for the cell to make proteins, and ricin is indeed a protein. It's from castor oil seeds and is very deadly to humans. The story there in the beginning of the chapter talks about Georgy Markov, who defected to England and was working as a journalist for the BBC. As he made his way to the bus stop on a London street, an assassin used the tip of a modified umbrella to jam a small ricin-laced ball into his leg, and Markov died in agony 3 days later. A little story there. And I know in the TV show Breaking Bad that they talked about using ricin as a poison there as well. There's a picture of the protein ricin right here. It's kind of a simpler protein. It's got 2 polypeptide chains, and let's see, one of the chains is an enzyme that inactivates ribosomes, and ribosomes are where your cells actually assemble all the proteins for your body. And the other polypeptide chain helps the ricin molecule across cell membranes, making it quite deadly. So this chapter details how cells convert information encoded in their DNA to an RNA, and then finally to a protein product. Cells need protein. Your body needs protein, and if you can't get those proteins, you soon die. And ricin inactivates those ribosomes that are important for making proteins. That's why it's such a deadly poison. DNA contains all the instructions for building a new individual. As we remember from last chapter, the order of the A, T, C's, and G's is also called your genetic code or your genome. We have a picture of a DNA molecule here, and I'll remind you that in DNA there's the 4 different nucleotides. We learned this last chapter. There's the thymine nucleotide, the adenine, the cytosine, and the guanine. Remember, each nucleotide has its base, a nitrogen-containing base. Then it has the sugar deoxyribose and the phosphate group, building a ladder molecule with the hydrogen bonds in the middle. A's always with T's, C's always connecting G's, and then you twist it to make the shape the double helix. And the order of these A, T, C's, and G's is what's called our genome or genetic code. All of our DNA is wound up on chromosomes, 46 of them to be exact for humans. And among those 46 chromosomes, segments of this stuff are called genes. We have between 20 and 30,000 genes to make a human. And those genes then code for usually a protein product, but can also code for an RNA as well. RNA stands for ribonucleic acid, and we'll look at how similar that is to DNA and how RNA is involved in making proteins. Again, what do genes code for? They code for proteins. cells make the proteins, and then those proteins build our structure, such as cells and body parts. This is also called the central dogma, this flow of information in a cell from the molecules of DNA. Then it's actually transcribed into what's called RNA. Then it's translated into a protein, and finally that protein is a trait or a physical characteristic that we have, such as the proteins that cause the colors of your eyes or the colors of your skin. Here it says DNA gets all the glory, but really, in reality, proteins do all the work. They're like the workhorses of the body. Here's another picture showing this. DNA is actually transcribed by mRNA, and ribosomes then are able to assemble proteins according to DNA's code. One thing to know is that DNA again is in the nucleus of a cell, and DNA is a big molecule. Remember, it's a double helix. So one problem is that since it's such a big molecule, it can't get out of the nucleus. But yet the structure that actually makes the proteins is the ribosome, and that's out here in the cytoplasm. So we need another molecule to act like a messenger to bring DNA's code out to the ribosome. And so that's where mRNA comes into play. M stands for messenger because it acts like a messenger. And you'll see how mRNA is designed. Instead of being a double helix like DNA, it's actually just a single strand which can fit through little pores of the nucleus and therefore can travel out into the cytoplasm to the ribosome. And therefore the ribosome locks onto it, and because mRNA copied DNA's code, it's like a messenger bringing DNA's code out here to the ribosome, and from that code we'll see how the ribosomes actually assemble proteins. Remember that proteins are actually chains of amino acids, and it's important for a protein to get that chain just right in the right order of amino acids in order for it to work properly. So the process called transcription is where we convert the information from a gene of DNA to RNA. It's important we do that because DNA stays in the nucleus and RNA is what will move out to the ribosome. Enzymes use the nucleotide sequence of a gene as a template to synthesize a strand of RNA, also called ribonucleic acid. Transcription, the process by which RNA is assembled from nucleotides using the base sequence of a gene as a template. There's 3 types of RNA that have a role in protein synthesis. There's ribosomal RNA, or rRNA. This is the main component of ribosomes themselves. These are the structures where proteins are built. Before a protein is complete, it's actually called a polypeptide chain. Once a polypeptide chain is put together, it undergoes some more processing before it actually becomes a mature protein. Transfer RNA, or tRNA, is also involved. tRNA is in charge of delivering amino acids to the ribosomes. And of course, there's mRNA, or messenger RNA. And messenger RNA actually makes a copy, or it transcribes DNA's code and carries the protein building message. RNA is a single-stranded chain of 4 kinds of nucleotides, just like DNA, but with a bit of a difference. Just like DNA, an RNA nucleotide would have its phosphate groups, would have a sugar. Instead, the sugar is called ribose, not deoxyribose. And thymine does not occur in RNA like it does in DNA. Instead, a very similar molecule called uracil occurs. So, the 4 bases of RNA would be A, G, C, and then U. And the U replaces the T. Adenine, cytosine, guanine, and uracil. Thymine is only found in DNA. Uracil is found in RNA. Translation, then, is a process that converts the information from the mRNA to an actual protein, first a polypeptide and then a protein. The mRNA carries a protein-building message encoded in the sequence of sets of 3 nucleotide bases. Those are called codons. mRNA is decoded or translated into a sequence of amino acids, resulting in a polypeptide chain that folds then into a protein. Transcription and translation are all part of gene expression, a process by which information encoded by a gene is converted into a structural or functional part of a cell or a body part. DNA to RNA to protein. Again, that's like the central dogma. So the sequence of amino acids in a polypeptide chain corresponds to a sequence of nucleotide bases in DNA called a gene. And the conversion of information in DNA to protein occurs in 2 steps, transcription and then translation. And we'll take a closer look at each of those. So remember, DNA, it's caught in the nucleus of the cell, but we need to get that message out to the ribosome, which is in the cytoplasm. So we need to go from DNA, nucleic acid language, to RNA, nucleic acid language, because RNA is going to act like the messenger to bring DNA's code out to the ribosome. Again, a reminder, RNA, or ribonucleic acid, has the sugar ribose. It does have, again, those nitrogen bases, C's and G's, cytosines and guanines, and then adenine, and instead of thymine, we'll find uracil. And the pairing is similar. C's will match with G's. A's, this time, will match with U's. RNA is only single-stranded, so it is able to fit through the nuclear pores and then travel out to the ribosomes. And there are quite a few different types of RNA to keep track of. The mRNA is what transcribes DNA's code in the nucleus and then travels out to the ribosome. tRNA, once again, brings the amino acids to the ribosome, or the rRNA, ribosomal RNA. And we won't worry about this one for right now. Here's a picture to compare. Here's DNA with its double helix. And here's RNA. Looks a little different, doesn't it? It's just single-stranded with its bases just hanging off here. The process of going from DNA to RNA is called transcription. During transcription, DNA acts like a template where RNA is assembled from RNA nucleotides. Each new RNA then will be complementary in sequence to the DNA template. And just with the old pairing rules, G pairs with C and A pairs with U. We need enzymes to help get this job done, so RNA polymerase, got that ASE ending there to show that it's an enzyme, adds nucleotides to the end of a growing transcript. Here's a picture to help visualize that. Here we have our molecule of DNA. It's been unzipped temporarily so that RNA polymerase can get in here and start adding molecules of RNA that match up with the molecules of DNA with the same pairing rules, except remember we've got to worry about that U. So if there's an A on DNA, then a U in RNA gets put in place. If there's a C in RNA, or excuse me, if there's a C in DNA, then a G in RNA gets put there. If there's a G in DNA, then there'll be a C put there with RNA. If there's an A, again, here we have a U, and if there's a T in DNA, we still use the same pairing rule that an A gets put in place. So this big pink thing here is the RNA polymerase enzyme that's helping to put these RNAs, these nucleotides, into place. And so we're building what's called a complementary copy of DNA, just one of the sides of DNA. Transcription begins with a gene on a chromosome. We don't have to transcribe all of your genome at one time. We only need to do a gene at a time as those proteins are needed. RNA polymerase and several other regulatory proteins will attach to the specific binding site called the promoter. That would be right here at the beginning of the gene. RNA polymerase then starts moving along the DNA over the gene, unwinding or unzipping the double helix to basically read the base sequence of the DNA strand. Then RNA polymerase bonds the free-floating nucleotides of RNA into the chain in the order dictated by that gene sequence, making an RNA copy of the gene, as we see back in this picture once again. When the polymerase reaches the end of that gene, the DNA and the new RNA strand are then released. Typically, many polymerases transcribe a particular gene region at the same time, so that many new RNA strands can be produced very quickly. Here's another picture of that to help visualize. Here's our free-floating nucleotides of RNA, and I know these are RNA because I see U floating here. Those are uracils. Here's our DNA strand on the bottom, and here's our RNA polymerase moving over the DNA strand, adding in the RNA nucleotides. Remember, if there's a T in DNA, then an A in RNA will match it. C's with G's and A's with U's. Remember, you won't find any T's in the RNA. Remember that RNA polymerase is an enzyme that helps to carry out transcription. There it is in the picture. And a promoter is in DNA. It's the sequence to which the RNA polymerase binds. It's the beginning right before a gene, the gene that we want to transcribe at the time, the gene that we need a protein made from. Eukaryotes modify their RNA inside the nucleus, then ship it to the cytoplasm. So basically, after an RNA molecule is transcribed, it's not ready to go quite yet. There has to be some modifications first. First of all, introns are nucleotide sequences that are removed from the new RNA. Introns are actually parts of DNA that don't code for anything, so they need to be removed from the RNA. Exons are going to be the sequences that actually code for something, so they need to stay. Sometimes some exons are removed and the remaining exons are spliced together. That's called alternative splicing, which then enables one gene to encode different proteins. So again, an intron is a nucleotide sequence that intervenes between exons and is taken out or excised during RNA processing. The exon is the sequence that is not spliced out, it's kept. It's needed because it actually codes for something. Sometimes they refer to introns as junk DNA because those don't code for anything. Alternative splicing is when RNA processing occurs and some exons are removed or joined in various combinations. Here's a picture that might help you visualize that a little bit better. Here's our eukaryotic DNA with both its introns and extrons. The exons are blue and the introns are orange here. Then we make our mRNA transcript and then we modify it. We splice out those introns so that we have just the exons left. So we may go from something very long of approximately 10,000 nucleotide bases to something only 1,000 nucleotide bases long by taking out the junk information. And we make our mature mRNA transcript. Again, the alternative splicing is when alternative mRNAs are produced from the same gene. So in this case, different segments can be treated as exons. Here the penguin's saying it's starting to get hard to define what a gene actually is. I agree with them. New transcripts that will become mRNAs are further modified after splicing. First of all, a modified guanine cap is added to the 5' end. This will help the mRNA bind to the ribosome. And a tail of about 50 to 300 adenines, or A's, is added to the 3' end. This is called the poly-A tail. Reason being is that in its journey from the nucleus out to the cytoplasm to the ribosome, enzymes and other cell molecules can degrade the mRNA. So by putting the 5' cap on one end and the poly-A tail on the other, it helps to preserve our mRNA. So again, transcription is going from DNA to RNA. During transcription, one strand of the DNA double helix serves as a template to assemble a single complementary strand of RNA called a transcript. Each transcript is an RNA copy of a gene, and we often call it mRNA or messenger RNA. Remember, there are 3 types of RNA that interact that will translate DNA's information into a protein. We have our messenger RNA. We just looked at how that's created. We'll also have our tRNA come into play and our ribosomal RNA come into play. Here's a picture of ribosomal RNA down here. Ribosomal RNA actually has 2 subunits that come together. They look kind of like blobs. The rRNA or ribosomal RNA are together what makes up a ribosome. And this is where our mRNA will meet, and also we haven't talked about it yet, the tRNA will meet here too. So this is the big meeting spot of all the RNAs, and cells have many ribosomes in order to make proteins. In ribosomal RNA, in a ribosome, you have an A site right here. This is what holds a tRNA, which carries an amino acid. A P site. This is what holds a tRNA carrying a growing polypeptide chain, and then an E site or exit site. This is an empty tRNA that leaves the ribosome. And we'll take a look at this next. So the protein building information in mRNA consists of a sequence of 3 mRNA bases. So each code for a particular amino acid. Remember, amino acids build proteins. So basically what this is saying is that we have our long mRNA strand, and every 3 letters is a codon that will code for an amino acid. Remember the choices in letters? We have A, C's, G's, and U's. And they can be combined into 64 different codons, which constitute the genetic code. For instance, AUG is a codon that codes for an amino acid called methionine, or MET for short. Instead of writing out these long amino acid names, we often just use the first 3 letters to symbolize that amino acid. UGG codes for an amino acid called tryptophan, or TRP. So once again, here's our DNA code. This was copied by mRNA to make a complementary copy, and every 3 letters codes for an amino acid. For instance, remember AUG codes for methionine. CGU, the next 3, code for arginine. GUA is the codon for valine. AAU codes for ASN. Can't remember off the top of my head what amino acid that is. GCA codes for alanine. UGC codes for cysteine, and GCC again for alanine. From just the 4 nucleotides in DNA and RNA, we can code for our 20 amino acids that make up our proteins of our body. Again, a codon in mRNA is a sequence of 3, or a triplet, that codes for an amino acid, or even a stop signal during translation. And the genetic code is the complete set of the 64 mRNA codons. There's only 20 kinds of amino acids found in proteins in the human body. So, some amino acids are specified by more than 1 codon. Some codons signal the beginning and end of a protein coding sequence. For instance, AUG, or methionine, is the start, always the start of translation. It's kind of like a signal to the ribosome that it will start making a protein. UAA, UAG, and UGA are the stop codons. Basically, that signals a ribosome to let go because the protein is done. The order of mRNA codons determines the order of amino acids in the polypeptide that will be translated from it. Remember, a polypeptide is kind of an unfinished protein. Here's the genetic code. Over here, you look up your first base, your second base, and your third. So for instance, let's say that your mRNA was CUG. So over here I would look up C, and then U, and then match it up with G. Here's CUG, so my amino acid would be leucine. And you can also see where the start codon is, AUG, methionine, and the 3 stop codons to let the ribosome know that the polypeptide or protein is done. Yes, the code is redundant. Several codons for each amino acid. That's alright. Like serine here has 4, and phenylalanine has 2. Glycine here has 4. Ribosomes, containing rRNA and structural proteins, and tRNAs interact to translate an mRNA into a polypeptide or protein. One large and one small ribosomal subunit join with the mRNA, and rRNA enzymatically catalyzes the formation of a peptide bond between amino acids, basically building the polypeptide chain or protein. Transfer RNAs are in charge of delivering the amino acids to the ribosomes in the order specified by mRNA. The transfer RNAs are floating around in the cytoplasm and arrive at the ribosome. Here's what a tRNA looks like. Looks a lot different than the mRNA, who's long and single-stranded, and the ribosomal RNA that looked like a bunch of blobs. This one actually looks like a clover leaf, because these 3 bases at the bottom here will match up with the codon on mRNA. This is called the anticodon. So the codons are on mRNA and the anticodons are on tRNA. So let's try this. If we know that an anticodon is AAG, what will be the mRNA that it will match with, the mRNA codon? Well, we know with the pairing of RNA that A's always go with U's. So it will be UUC will be the codon that matches with this anticodon AAG. Here's a couple other looks. Sometimes they diagram the tRNA to look like this, and sometimes they diagram it to look like this. On the end here, this is the site that grabs a hold of the amino acid and kind of like grabbing it by the hand and bringing it to the ribosome. So it kind of looks like this. Here's our DNA's code and mRNA, which transcribe the mRNA, which transcribe the DNA, every 3 letters being a codon, coding for an amino acid. And it's just routine that when we look up amino acids, we use the mRNA. So here I would look up AUG, and in your book they have a chart. Your chart is on page 142. So you would look up AUG and you would find it would belong to the amino acid methionine. Here's our tRNA here, the anticodon UAC, complementary to AUG. Kind of like, again, grabbing MET by the hand and bringing it to the ribosome. Each of these coming in order according to mRNA, which, remember, came from the original code of DNA. Each tRNA, again, has 2 attachment sites. One side, the anticodon, and the other side, which grabs the amino acid. So remember, with RNA, there's your different kinds. The mRNA, it carries DNA's protein building instructions. Its nucleotide sequence is read 3 bases at a time, called codons. 64 mRNA-based triplets or codons represent the genetic code, and there's 2 other types of RNA. Don't forget about tRNA and ribosomal or rRNA. Translation converts the information carried by an mRNA into a polypeptide or protein. It occurs in the cytoplasm at the ribosome 3 stages: initiation, elongation, and termination. So in initiation, when we're trying to build a polypeptide or protein, initiation brings together the mRNA and the tRNA. This big brown blob here is the ribosome, or the ribosomal RNA. And these little blue things are the tRNA bringing an amino acid. And the pink thing, long strand here, is the mRNA. So see how the ribosome reads the mRNA code every 3 letters and then matches the codon on the mRNA up with the anticodon on the tRNA, and then grabs onto the amino acid that the tRNA brought, and then strings those together in the proper order. Elongation is adding amino acids, and then termination is the end codon. Remember, we're going from nucleic acid language here to amino acid language, trying to get the proper order of amino acids so we make the right protein so it works properly. And again, that's called translation. It's an energy-intensive process by which a sequence of codons and mRNA is converted to a sequence of amino acids in a polypeptide chain, which then will be made into a mature protein, and these proteins will build your body. tRNAs deliver amino acids to ribosomes, which catalyze the formation of peptide bonds between the amino acids. So, can you look at this picture and tell the story from the beginning? mRNA making a complementary copy of DNA's code, then getting modified, splicing out the introns, adding a guanine cap and a poly A tail, going through the nuclear pore, out to the ribosome, where it's actually read in the order of each the codons every 3 letters, which then a tRNA brings an amino acid, and they are then strung together in the proper order to make a polypeptide, which will then later on become a mature protein. If a mutation occurs, changes in the genetic instructions can happen, and you'll get an altered gene product or an altered protein. It could be neutral, it could be good, and it might be something that is lethal as well and doesn't work. Hemoglobin is a great example. Hemoglobin has 4 polypeptides folded around an iron-containing cofactor, and various defects in the polypeptides can cause anemia. Here's a picture of hemoglobin with its 4 polypeptide chains, its 4 components. Different mutations can occur. A deletion from DNA of the beta globin gene causes a type of anemia. A deletion is where you lose one or more of the bases that are in DNA. An insertion, it's a mutation, can alter polypeptides, is where you actually have bases added to DNA. So the names really do say what they do. A deletion deletes, an insertion inserts. Taking out letters if you're a deletion, adding letters if you're an insertion. This becomes a frameshift mutation. So if you take out letters, it's going to shift your code. If you add letters, it's going to shift your code again, and this can therefore change your order of the genetic code, therefore changing the order of mRNAs, and therefore changing the order of amino acids and changing your protein product. Other types of mutations that do not cause frame shifts in a base pair substitution, kind of just what it says it does, a base gets substituted. And here's a great example that involves hemoglobin. It's called sickle cell anemia, and it's a substitute of valine for glutamic acid. This causes the hemoglobin protein to clump. Normally, red blood cells should be round, but this mutation causes red blood cells to be sickle or banana shaped. Here's a picture of what that looks like. Here's a normal red blood cell, nice and round and pretty, but a sickle cell with the mutated hemoglobin causes the sickling or the banana shaped hemoglobin, making the red blood cells become this shape. Just one amino acid exchanged for something else, exchanged for valine, causes this to happen. So out of all 146 amino acids that make hemoglobin, just one can have this devastating effect. There's many causes of mutations, like transposable elements can cause insertion mutations. Mistakes can occur with DNA replication. Environmental agents like UV light, pollutants and contaminants can cause it. Other chemicals can cause mutations. Ionizing radiation, such as X-rays, can break chromosomes into pieces. UV light can form thymine dimers. This is something that causes two thymines next to each other to kind of bond with each other, and that kinks your DNA, and that's not good. Chemicals in cigarette smoke can actually cause mispairing during replication, or even stop replication entirely. Those are all environmental factors that can cause mutation. So a mutation is a small-scale, permanent change to the nucleotide sequence of DNA that can result from replication errors or from the activity of transposable elements or exposure to other environmental hazards. Such mutations can change a gene's product, can change those proteins that you so desperately need for your body to function correctly. Sometimes mutations are as bad as that. Sometimes they are neutral, and sometimes they can be good and beneficial. So back to ricin, a modified form of ricin. Remember, ricin is a protein, is currently being tested as a treatment for some types of cancer. Ricin is attached to an antibody that can find cancer cells in a person's body. And researchers hope that the attached ricin will kill the cancer cells without harming the normal ones. And this could be a possible treatment for cancer in the future. Once again, be sure that you read the chapter and take a look at the questions at the end of the chapter as well. Critical thinking and the self-quizzes, the data analysis activities, and the summary back there at the end of the chapter.